Hello everyone, I am Dr. Chethan, working as an assistant professor at the Department of Dermatology at Sri Lakshminarayana Institute of Medical Sciences in Pondicherry. Today, I will be covering a topic called as basic site lab investigations. So the site lab investigations are the very simple tests which we do in the bedside or in the OPD setting, which will aid in the diagnosis of the patient, especially when the lesion is not very clear, just by clinical examination. And these tests will consume less time, and they are very cost effective, and they are very easy to perform. So these are the various side lab investigations which we use in dermatology. So it includes gram staining, acid fast staining, slit skin smear, Zang smear, tissue smear, potassium hydroxide mount, wet mount of genital secretions, dark field examination, wood slab, hair examination, and sometimes even urine stool and hematological investigations such as PSR. So gram staining, as you already know, it's a differential stain. That is, the gram positive organisms will stain differently when compared to gram negative organisms. So this differential staining happens because of the differences in the cell wall constituents. The gram positive organisms are made up of 60 to 90% peptidoglycan molecules and decoic acid molecules, whereas gram negative organisms, the cell wall in gram negative organisms, the cell wall is made up of only 10 to 20% of peptidoglycan and decoic acid, and instead there will be more amount of phospholipids, polysaccharides and lipoproteins in the gram negative organisms. So that is why some organisms will take up one stain and the other one will not take up that particular stain. So that will lead to the differential stain. So the basic dye which is used in gram staining is methyl violet. The mordant which is used is gram iodine. The decolorizing agent which is used is 95% ethanol, and the counter stain which is used is dilute carbonfusion or 1% saffronin or even weak neutral red. So the fixation of the slide will be by heating. So this is the gram staining procedure. First you have to pour the methyl violet on the slide and you have to wait for 1 minute and then rinse the slide, then add gram iodine for 1 minute and then rinse the slide with water and then add 95% alcohol drop by drop. Again rinse with water and add the counter stain which is dilute carbonfusion and wait for 30 seconds to 1 minute. So this is how Staphylococcus aureus looks like in gram staining. So you can see the purple colored or gram positive organisms which are located in a cluster or grape-like appearance. So this is very typical of Staphylococcus aureus. So this is particularly important when patients present with bacterial infections such as folliculitis in the skin. So this is how a gram negative organism looks like. It almost appears pink in color. So one typical example is a Neisseria gonorrhoeae. You can see there are multiple intracellular diplococci which are located inside this cell. So these are gram negative organisms. So this is very useful in the diagnosis of gonococcal urethritis which might present to our skin infection. So this is chancroid, which is a gram negative rod. So you can see that the organisms are like arranged in a linear line. So this is called as railroad track appearance or school of fish appearance, which is seen with Haemophilus eucrei, and it is very useful for diagnosis of the chancroid. So typical examples, gram positive organisms include, for gram positive cocci, we have Staphylococcus aureus, which will be present in clusters, and Streptococcus will be present in chains. If there are in pairs, you should suspect Streptococcus pneumoniae. And among the gram-positive bacilli, we have Bacillus anthracis and Clostridium species. And gram-negative cocci, we have Neisseria gonorrhoeae and Neisseria meningitidis. And gram-negative bacilli includes E. coli and Klebsiella pneumoniae. Next, we'll look at acid-fast staining. Acid-fast staining was developed by Franz Zeel and Frederick Nielsen. So this is again a differential staining, and the main principle here is that the presence of unsaponifiable wax fraction called as mycolic acid in the cell wall of these organisms. So this mycolic acid is the presence of mycolic acid is the most important reason by which we have come up with acid-fast staining. So the reagents include the primary stain such as strong carbon-fission. The decolorizing agent is 20% sulfuric acid or 3% acid alcohol. The counter stain is low fluoresce methylene blue. So fixation will be alcohol fixation using methanol for 2 to 3 minutes. So first you have to add strong carbon fission, wait for 5 minutes with intermittent heating, then rinse the slide, then add 20% sulfuric acid or 3% acid alcohol mixture and wait for 10 minutes, and then rinse the slide again and add low fluoresce methylene blue and wait for 15 to 20 seconds. So if at all for skin, how is the acid-fast staining useful? So in skin, the main acid-fast bacilli which we will encounter is Lepra or Hansen's bacillus. So this one will be, if at all if there is positivity, the slit skin smear. If the acid-fast staining after a slit skin smear is positive, we will call it as multibacillary leprosy. And multibacillary leprosy differs from paucibacillary leprosy in that the multibacillary leprosy will have a higher amount of bacterial load and requires a longer duration of treatment time. So that is why it is very important to do acid-fast staining in a case of leprosy. So this was the scientist who discovered Hansen's bacillus, Gerard Arnold Hansen. So it's the thing. And for slit skin smear, we generally use both the earlobes and 4 representative active skin lesions. But now the WHO guidelines have changed, and only one side of the earlobe, and along with that, we are going to use the representative active skin lesion as a site. So instead of 6 lesions, in 6 sites, we are going to do only 2 sites these days. And the materials used are these ones. You need a spirit, you need a spirit lamp, then you need slides, you need cover slips, and then you need forceps, and you need spirit. So these are the things which are required. And along with that, for staining, you require carbon fission, 5% sulfuric acid, and low fluoresce methylene blue. So the procedure I have already explained. So this is the procedure of slit skin smear. You can see that a cut is made in the earlobe, and that will be smeared on the slide. It will be fixed, and then we will add carbon fission, 5 minutes with intermittent heating. Rinse the slide, add 5% sulfuric acid or 1% acid alcohol, wait for 10 minutes, then rinse again, add low fluoresce methylene blue for 15 to 20 seconds. So how this particular procedure differs from the earlier procedure which I mentioned is by the concentration of the sulfuric acid which is used here. So as we already know, the tuberculous bacilli is also a acid-fast bacilli, whereas the difference here is tuberculous bacilli is actually, the TB bacilli is actually more acid-fast when compared to leprosy bacilli. So when you are suspecting tuberculosis, you will ideally use 20% sulfuric acid, whereas when you are suspecting leprosy, you should add 5% sulfuric acid for as a decolorizer. So these pink elements which are present here are called as acid-fast bacilli. So they are a little bit curved or straight pink rods which will be present. and sometimes they can cluster together to form some elements called as loci. You can see there are multiple loci in the pink colored loci which are present. So this is a case of Mycobacterium leprae, and you can also see individual red staining or pink staining elements here. So they are again individual Mycobacterium leprae bacilli. So we'll have something called as Ridley's logarithmic scale in order to report a slip skin smear. And we also have bacteriological index and morphological index, which we can use to report a slip skin smear slide. So next we'll look at Zhang smear. So Zhang smear was introduced by Arnold Zhang, and this is a very useful test in the diagnosis of various viral infections of the skin. So first we look at the collection of specimen, especially viral diseases such as genital herpes or chickenpox. So you have to uh, take a vesicle which is newer, that is with a vesicle which is less than 72 hours old, or you can even take ulcer if there are no active vesicles. Then first you have to rupture the vesicle, you have to deroof the vesicle, and from the floor you have to take the smear. So whereas in case of herpes, you can even use a cervical swab, and even you can collect the vesical fluid itself for doing a smear. And in case of molluscum contagiosum virus, you can collect the cheesy material which is expressed out of the molluscum lesions. And in case of ulcerated tumors, you have to first remove the crust, and for non-ulcerated tumors, you have to put a superficial incision and then make a smear out of it. So the stain which is commonly used is Giemsa stain. You can also use Wright stain, Leishman stain, HND, methylene blue, all those things you can use for doing a Zhang smear. The most commonly used ones are Giemsa stain and Leishman stain. So Giemsa stain contains using azure B and methylene blue, and Leishman stain contains polychrome methylene blue, using azure, Leishman powder, acetone free, absolute alcohol. So this is the procedure of Zhang smear. First you have to air dry the slide, and then mix it with methanol, and then add with Giemsa stain and wait for 15 minutes, and then air dry the slide and examine under oil immersion microscope. In case of Leishman stain, you can do it with a similar manner. So first you have to air dry the slide, then cover it with the Leishman stain for about 2 minutes, and then add double volume distilled water and wait for 15 minutes, wait for 5 minutes, and air dry the slide and examine under oil immersion microscope. So what you are going to see after doing this smear. So basically what you are going to see is, you are going to see this in case of autoimmune disorders such as pemphigus vulgaris, you are going to see these acantholytic cells. So this round colored, these round shaped cells, which are like which have a round nucleus inside them. So these are called as acantholytic cells. So these are nothing but the keratinocytes which are separated from the rest of the surrounding keratinocytes. So these are acantholytic cells. This is typically seen with autoimmune blistering disorders such as pemphigus vulgaris. In case of herpes zoster and herpes simplex, you will see something called as multinucleate giant cell. You see that the multiple cells are fused together to form a, it appears as though they have formed a single cell here, and you can see there are multiple nuclei which appear close together. So this itself is called as multinucleate giant cell. Here again there is a multinucleate giant cell which is seen in herpes zoster. So other than autoimmune blistering disorders and viral infections, Zhang smear is also useful for the diagnosis of molluscum contagiosum, bullous impetigo, staphylococcus pallid skin syndrome, toxic epidermal necrolysis, basal cell carcinoma, and even squamous cell carcinoma. So then we'll see the tissue smear. So tissue smear is something which is used for diagnosis of donovonosis. So donovonosis, as you already know, so donovonosis is a disorder of the genitalia. It is a commonly a sexually transmitted disease, which will present with the granulomatous ulcer, so which will bleed on touch. So in this particular tissue smear, what we are going to do is we will cut a tissue from the active edge of the granulomatous ulcer using a tooth forceps. And this particular tissue is placed between two slides, and the slides are brought together, and the tissue is crushed between the two slides. So this particular slide is then hair dried and fixed and stained with either Giemsa stain or Wright stain, and it will be examined under oil immersion microscope. So this is the tissue smear. You can see here there are multiple gram positive organisms which are present inside the cell here. So this is these are inclusion bodies which are seen in Klebsiella granulomatous called as Donovan bodies. So next we'll look at the potassium hydroxide mount. So potassium hydroxide mount is a test which is used for demonstration of fungus or yeast from the skin lesions. So for collecting the sample, first we have to do a scraping. So you can either scrape from the skin, you can scrape from the hair, or even from the nail. So from skin scrapings, you should generally take from the scaly areas, that is at the border of the lesions. And for hair, you can take scrapings, or you can even take the entire hair, cut it into smaller stubs and crush, and then examine it under microscope. Or for nails, you can take deeper nail scrapings or from the nail clippings, or you can avulse the whole nail and then examine with potassium hydroxide mount using a microscope. And regarding the genitalia, especially for women with white discharge, you can collect the discharge directly from either the posterior fornix and skin surrounding the genitalia. And from men, you can collect it from the surface of the glands with using a Western swab. So then, after collecting the specimen, you have to mount the specimen to the slide. So the specimen is mixed with a drop of 10 to 20% KOH on a glass slide, and it is covered with a cover slip. So generally, for scrapings from the skin, it will take around 10 to 15 minutes for the digestion and clearing. And for hairs and thick hyperkeratotic specimens, it will take anywhere from 30 minutes to 2 hours. And for nails, it will take anywhere from 24 to 48 hours using 10% KOH. So one important thing is what the KOH does is it will basically digest the cellular debris which will be present in the scrapings. So once the cellular debris is cleared, you will be able to see the fungus more clearly. So that is the basic principle here. So to do the microscopic examination, initially you will do under low power and low intensity of light, and later you will do with higher magnification, that is at 40x with higher illumination. So this is you can see there are, this is the potassium hydroxide mount of a teniocarpus patient. So you can see that there are multiple reflective elements. You can see there are multiple hyphae, the long structures which are present, thread-like structures which are present in this slide. So they are almost transparent. They are reflectile substances. So they are something which is common with teniocarpus. So this is a dermatophytic infection or teniocarpus. So this is a slide showing spaghetti and meatball appearance of Malassezia purpura. So Malassezia purpura has these thread-like structures called hyphae, and along with that there are spores which appear round in shape. So since there is both these round-shaped structures as well as hyphae, we will call it as spaghetti and meatball appearance or banana and grapes appearance, which is seen in Malassezia purpura, which causes pityriasis versicolor. Then regarding candidal infections, we have something called as yeast cells which will be present here. You can also consider doing a whiff test for the diagnosis of candidiasis, and then we also have something called as scabies. You can also do potassium hydroxide mount for scabies. So in scabies, basically the surrounding debris will be dissolved, and because of that, the mite will become mite, or fecal element will become prominent. Whip test is useful for diagnosing bacterial vaginosis, not for candidiasis. So then we have something called as wet mount examination. So here, instead of using the potassium hydroxide, we use something called as normal saline or distilled water. So in men, we will collect the sample from the urethra. Again, in women, we will collect the sample from the posterior pharynx. So the collection is the discharge is then mixed with normal saline on the slide, and it will be covered with a cover slip. So what we are basically trying to diagnose is Trichomonas vaginitis, so which will be caused by the parasite called as Trichomonas vaginalis. So you can see that these are the parasitic elements which will be seen under wet mount examination. And in case of bacterial vaginosis, we will see something called as clue cells. So the clue cells are nothing but vaginal epithelial cells with multiple attached bacilli to that called as borderline bacilli. So these are the clue cells which are seen in bacterial vaginosis. So dark brown microscopy is again something which is useful for the diagnosis of syphilis. So although syphilis was not very common for a few years, now because of the emergence of HIV as a pandemic, syphilis is actually coming back to the community in a larger number, and the specimen is collected from the directly from the lesions, and here the collection of this dark brown microscopy is actually most productive in the syphilis when it is in the primary or secondary stage, and in case of early congenital syphilis. So under aseptic precautions, you have to clean the lesion with normal saline, and if it is crusted or contaminated, you have to remove the crust and wipe away any blood-stained exudate from the using a dry gauze and apply pressure till a clear serum exudates from the lesion. So this serum should be directly collected on the glass slide or a cover slip, and then cover the edges of the cover slip with a Vaseline. So when you examine under dark brown microscopy, what you are going to see is called as motile spirochetes called as Treponema pallidum. So you can see that they are having a corkscrew structure. So only the background is lighted up, whereas the organism is appearing bright. So that is why it is called as dark brown microscopy. So these organisms will be motile. So we have a simple examination tool called as Wood's lamp. So Wood's lamp was invented by Robert W. Wood. It is a high pressure mercury arc fitted with a compound filter made up of barium silicate with 9% nickel oxide. So the principle is this particular lamp emits a ultraviolet radiation which will peak at 365 nanometer, and this UV light is absorbed, and visible light is emitted from wherever this particular UV light, this thing is shine. So in compatible lesions, so the UV light is absorbed and visible light is emitted. So that means the lesion in question will appear brighter when compared to normal skin. So the prerequisites for doing a Wood's lamp examination include the dark room, because no other visible light should be present. Only it should be coming from the lesion. So that is why we should have a dark room. We should have a dark room. And first we should warm up for the 1 minute. That is, after turning off the light, you should leave the patient alone there for about 1 minute. And you should avoid washing the lesion. And the light source should be held up around 4 to 5 inches above the lesion. So it is useful in the diagnosis of superficial fungal infections, such as Microsporum species, which will emit blue-green fluorescence, Trichophyton shown lining, which will emit dull blue fluorescence, and Malassezia purpura, which emits yellow fluorescence. So certain bacterial infections such as Pseudomonas aeruginosa emits green fluorescence, and Corynebacterium magnesia and Propionibacterium acnes will emit fluorescence, and even there can be we can also elicit the burrows in case of scabies. So this is a good slab examination of tinea capitis caused by a microsporum species. You can see this blue-green kind of fluorescence which is coming out of the skull. So this is a case of erythrasma which is caused by Corynebacterium PCM. So you can see the coral red color fluorescence which is seen after shaving blue slab. So it is also useful for diagnosing certain hypopigmentary and depigmentary dermatosis. So in vitiligo, the lesion appears bright blue-white. So this will also help in differentiating between lesions such as nevus depigmentosus and nevus anemicus, and it is also useful for differentiating chemical leukoderma, active macules, and hypopigmentosis. And in case of hyperpigmentary dermatosis such as melasma, we can tell the depth of involvement. That is, we can tell an epidermal melasma from the dermal melasma using a post-lab examination. And porphyrias can also be diagnosed using post-lab examination, where the porphyrias will get red-pink fluorescence from the involved structures. So this is the hair examination. Hair can also be mounted with cavage. So for a cavage mount for tinea capitis, you can see the spores are present in the outer layers, which is called as ectothrix. And when the spores are involved in the inner layer of the hair, it is called as endothrix. So by looking at the color, for example, certain pigment dilution disorders such as albinism, geriatric syndrome, and Griselli syndrome. So there will be decrease in the amount of melanin in the hair. So this is called as pigmental dilution. So that again is a helpful modality. And the shaft disorders such as monolithics and palatote can also be diagnosed using the hair examination. And polarized microscopy is especially useful for diagnosing trichothiodystrophy, where you will see something called as tiger trail bands, where there is alternate dark and lighter regions which will be present in the hair. So this is called as tiger tail appearance, which is seen in trichothiodystrophy. So other investigations such as urine microscopy, stool microscopy, and even ESR also can be done. So ESR is particularly useful for patient with in whom we are suspecting connective tissue disorders such as SLE and scleroderma to see the activity of the lesion, activity of the disease. So that is why. So some people will keep the ESR as well as urine and stool microscopy as a side lab investigation. So, thank you.